you want to see more in-depth things, or if you want to see images or pictures or things like that, then I can pull those up. How does that sound?
That sounds good.
All right. Yay. So is this the Ohana crew here? Awesome.
It's Ohana and Sunrise.
Yeah.
Oh, fantastic. Good, good, good. So I see some familiar faces. Welcome back, and welcome to new faces too. My name is Tara Chestnut, and I am one of the ecologists here in the park, and I've been here for about 5 years now. And the main sort of taxa that I work with here in the park are birds and mammals, although my academic specialty and background is in aquatic ecology, and I've spent over 20 years working with amphibians, and so I know a lot about them. And then I'm also the park research coordinator, so anyone that is from a university or from another public agency or from a nonprofit group who's interested in doing research in the park has to submit an application for a permit to do that work, and then I facilitate and coordinate the review amongst all of the different work groups. And I also serve on the federal steering committee for a nonprofit organization called the Partners for Amphibian and Reptile Conservation Park. And so there are a bunch of different hats that I wear in the park. And so the sort of foundation of my program is threatened and endangered species monitoring. And spotted owls are the species that we monitor most. We have a crew of 6 people that have been working since April doing surveys, and I think most people are familiar with spotted owls. Yeah. And with the conservation issues that are surrounding them, they were listed in the 80s and under the Endangered Species Act as a threatened species, primarily due to the way that we managed our forests and timberlands. And in the early 90s, law was passed, or I should say a policy was passed called the Northwest Forest Plan. And it served as a recovery plan for not just northern spotted owls, but also identified other species on federal lands that were a priority for research and monitoring. A lot of those species, we just didn't know a lot about them. They were weird things like mollusks and lichens and things like that. And so that law passed, or that policy passed, around the time that I was starting my career, and it was a really great time to be starting your career in natural resources. And what that policy also did was define 11 different study areas throughout the range of the northern spotted owl that we monitor in the same ways, so that we can learn about their populations at multiple different scales. And every 5 years, each of those 11 demographic study areas gets together to analyze the data. And we were supposed to do that last year. That was our 5-year, but the government shutdown prevented that from happening. And we had our meeting in January, shortly before COVID times happened. And so we were able to at least get started on that analysis. And, what we're finding is that, it's bad news for spotted owls, especially in the northern parts of their range. We had one of our technicians worked on her master's in, she finished that just 2 years ago, analyzing the data just for Mount Rainier National Park. And so we took it a little bit to a smaller scale than is covered in the demographic study. And what she found is that in spite of the fact that we have the most ideal habitat for northern spotted owls, that in the last 25 years that they have declined in the park by 50%. And that's that's a pretty massive decline, but it oddly feels like good news when in our whole demographic study area, the decline in the last 25 years is closer to 84%. So, yeah, it's not looking good for spotted owls. And the primary reason is because barred owls have effectively expanded their range from the east to the west because of, again, the way that we've managed our lands. Back sort of before European colonization, there weren't a lot of trees in the Great Plains. They were the Great Plains. And when it was colonized by Europeans, they brought with them houses and trees and things like that that allowed barred owls to expand their range. And so they've arrived in Washington and Oregon. They're slowly, or I should say quickly, invading into California as well. And there are some demographic study areas that are doing experimental barred owl removals to see if that's a tool that we can use to help support spotted owl recovery. And so that's not something that we're considering here at the park, but northeast of us in Cle Elum, they are doing that, and also at some sites in California. And so that's kind of where we are with spotted owls. And then another area of my program involves carnivores. And so we have several carnivores on our landscape, but only a handful that I actively sort of monitor. And so we have, many of you are familiar with our Fisher Reintroduction Program. Again, thanks to the timing of COVID times, we were able to just barely finish all of our translocations from Canada. And so we had the last release of the project in January. And there's lots of information on our website and also on the interwebs about that. We've got a lot of great media coverage where you can find information if you are interested in developing programs about that. And so we released all of the animals for both the North and the South. And the place where we are now is kind of waiting for them to do their thing. And we have funding applications in to monitor the next phase of Fisher recovery, which is seeing if they are reproducing enough and surviving enough so that they are on a path to having self-sustaining populations. On the Olympic Peninsula, they've finished that stage of their monitoring also, and what they were able to determine through genetic analyses is that they do need to augment the fishers on the Olympic Peninsula. And we're hopeful that we won't need to do that in the Cascades. And I feel optimistic about that because one of the things that we did was adapt our release strategies based on how we saw fishers move once they were released. And so on the peninsula, they kind of like peppered them all over the place and they traveled really long distances. And instead of doing that in the Cascades, one of the strategies that we employed was just to release them in a handful of sites and then let them disperse on their own. And that seems to have reduced their long distance movements. And they also seem to set up territories a lot sooner than animals on the Olympic Peninsula. So we're optimistic and we're hopeful. So that's kind of where we are with fishers. And the other two carnivores that we monitor are both alpine carnivores, Cascade red fox, and wolverines. And the person that we work with, or the organization that we work with to do a lot of that work, is the Cascade Carnivore Project, who is led by Jocelyn Akins. And the projects that she's working on right now, or that we're working on right now, are to estimate the number of Cascade red fox that are in the park, so that we can have a better understanding of their genetic diversity and and their movements between the northern and southern parts of their existing range. Foxes are not typically a species that are on people's radar, because they're all over the place. But the important thing is that the Cascade red fox, it's a montane fox that is only occurs in Washington, and all of the lowland foxes that we see in the cities and on, like San Juan Island and places like that, they are all introduced. And they were introduced for fur farming, and also just for, I don't know, British style hunting. And and so they're not native. So far, there's no indication that they're mixing, which is good. But what we've seen in the Sierras is that when mixing did occur with introduced foxes, it happened very quickly. And so we're concerned about their genetic integrity, because they have an effective population size range wide, which is basically like the number of females that are having kits every year is less than 20. And so that's not, when you think about just a population standpoint, you need more than 20 moms in the world at any given year. And then Wolverines, we have some really exciting news about Wolverines. It's sort of the opposite of what we're seeing with fishers and with Cascade red fox, is that Wolverines seem to be recolonizing their historic range in Washington on their own. And by some, I don't know who is looking after me and our funding proposals, but last year was the first year that we were able to get some substantial funding to monitor wolverines in the park, after a handful of incidental sightings. And we've confirmed that in the last 3 or 4 years, we've had at least 4 different individual wolverines in the park. And this year, we confirmed that we have a lactating female. So we have wolverine babies in the park, which is a really big deal. That's only the second den that has been documented in the South Cascades, in modern times. I did a search of all of the park records for wildlife, and there are no historic records in our collections of wolverines from the park. I'm sure that they were here, but they weren't. They certainly weren't common, and they weren't common enough to collect by any means. And so that's a really big deal. We're working on a press release, and then the other thing that Annie is helping me with is a carnivore tracking guide. And so it's something that we'll post to our website so that visitors and staff alike can have better tools to recognize the different carnivore tracks that are out on the landscape and have good solid instructions on how to document them so that they can be uploaded to our wildlife observations database. And we can share that with other partners like the Cascade Carnivore Project, because there's nothing more frustrating to, like, I think a visitor and resource manager to like have a report and then just have it not be documented the best way that it could be. And so we want to set people up for success, and we want to set people up for to contribute as best they can. And so I'm super excited about having that as a tool. And the other thing that I'm working with Jocelyn on is typically we have Cascade Red Fox survey training in August, and it's in person. It's usually limited to 11 people because of group size restrictions with wilderness. And instead of doing that group training, we're pivoting to have to make a training video that will outline all of the steps for how people can recognize fox scats and collect the samples and do that successfully in the park and outside of the park. And so it'll also make it more accessible to more people, which is super exciting. And so we're planning to film that in June. I guess it is June, at the end of June, and then and hopefully have the production team deliver it by the beginning of August, which is fairly ambitious, but I have great faith in them. And it's the same production team that made our Fisher video that I think most people have probably seen by now. It's really, really well done and beautiful, I think. What else? What else do I do?
You do bats.
Oh my God, I do bats. I've been on a bat call all day long. So that's right. That's why I was a little bit late. There's a national white-nosed webinar, and it's been going since 7:30 this morning, and my brain is a little zoomed out. But so in 2016, white nose syndrome was detected in Washington, and it was 1,300 miles from the nearest detection, which was sort of near Nebraska. And so it made a really big jump that a lot of people didn't anticipate. How it got here, we don't know. And in a lot of ways, it doesn't matter because it's here. And so what we've been doing in the North Coast Cascades network is combining all of the different tools that we have to monitor bats so that we can have a better understanding of their populations. And we do that by doing acoustic monitoring on the landscape, and then also doing colony counts in places where we have known bat colonies, which unfortunately is pretty much in just buildings and structures. And so it's really convenient for me that they're there, but it's also super gross because you don't want animals in buildings. Like, they poop and pee in there, and that's gross. And and it also just creates a worker safety hazard. Most, like, on the landscape, some of the most conservative estimates that I've seen are that less than 1% of bats have rabies, but behaviorally, a rabies infection changes animals, and it is more likely that you will encounter a bat if it's rabid. And so we don't want those in our workspaces and in our homes. And their poo and pee also carries histoplasmosis, which is a fatal lung disease, and we certainly don't want that either, especially right now. so so we do collect guano at our bat colonies so that we can screen them for white nose syndrome. Well, it's technically you're screening for the pathogen that causes white nose syndrome, but you can't actually diagnose an infection unless you see clinical signs of the disease. And so we can know that at least there are bats in that population that are carriers. And this season, we had to stand down our white nose surveillance because there was too much uncertainty around whether or not we could unintentionally transmit COVID-19 to bats. And so rather than taking that risk, we stood down our surveillance, which is, I think, good because, no one wants to be that guy. But the the risk, I think, is fairly low. But just to have an overabundance of caution, we are standing down all of our bat handling activities until we have more information and more certainty about what that looks like. So we did have 4 bats at our Ohana colony that tested positive for the fungus that causes white nose syndrome in 2017. But we did not see any bats that had clinical signs of the disease. And so we don't know if they were carriers or if they were just recovering from the infection and were already healed. But we haven't seen any clinical signs of the disease in that colony, and we haven't had any positive detections since then. So that's encouraging. And Michael Hansen, who is our bat crew lead this season, she is going to be starting her master's in the fall at Oregon State University, focusing on the colony at Ohana so that we can try to find where they're hibernating, which will help us get more information about sort of what the risk is. Because if they're hibernating in small places with just an individual or 1 or 2 other buddies, then the chances of them spreading it is much lower than if they were in a really big colony with hundreds of bats. And so we don't know where very many hibernacula are in our parks. And so it's kind of like a needle in a haystack. But some of the other tools that we're going to be using to support that are trained detection dogs that last season when we were doing our white nose surveillance, we gave some of the bats little tiny haircuts. And there is Jack is the dog and Colette is the handler. Colette trained Jack to identify bats by using their hair. And it's pretty remarkable what Jack has been able to do. He's been able to tell Colette where bats have simply landed on rocks for moments before they crawl into a hibernacula, and so it's seeming to be very promising as a technique. So we're excited about that. And then we're also planning to use radio telemetry, but you can imagine like the battery on a radio that goes on a bat is really, 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 really tiny. And so the batteries don't last that long. And so using the dogs to help us will allow us to be more productive in that work. So, yeah. I can't believe I forgot about bats. So if you live at Ohana and you want to help us out by doing emergence counts, we would love to have that help. It is a struggle for a lot of people, especially in June, because it's late by the time they come out. Um, but if you don't mind staying up past dark, we would be glad to have any help. It's super easy. You just stand there with a clicker and count them.
Should they email Michael or you or both?
Both of us would be great.
Okay, I'm gonna put you both on the chat.
Okay.
Cool. Does anyone have any questions for Tara? Wildlife related?
Okay, well, it's unmuted.
I mean, this is just like, I'm very personally interested in how you give the bats haircuts.
With little tiny scissors, like they were. So we used hemostats to kind of like pull their hair up and make sure that we weren't going to cut their skin. And then they were just like cuticle scissors that we just like deep, deep, deep. And it was just the tiniest amount. And Colette was like, I mean, it was like, I'm trying to like think of what is like, it was like the size of the top of a Lego, like one of those little, like a small Lego, like the round parts that click together. It was just like that much hair.
We're very overwhelmed with how cute that is.
It's adorable. It's so adorable.
I have a short question and a long question. The short question is, I heard an owl last night, and I went on Merlin Bird ID, and the closest one I heard was a great horned owl. Do we have those there?
I think the most common owl that we hear, yes, are barred owls, and I'm not particularly fond of their call because I like to cook, and their call is often described as them asking, "Who cooks for you?" And I'm like, "I cook for me."
And then the other question is there work being done for beaver reintroduction?
Say that again, I'm sorry.
Beaver reintroduction in park or area.
Beaver reintroduction. I don't think that the beavers are at a place where that's warranted. They're not they're not a species that has status, in that they're not threatened or endangered, and and we have beavers in the park.
Really?
But I think in other places, they've definitely suffered some big losses, and I think that in other parts of the country they've been able to do incredible restoration work by bringing back beavers.
Yeah, because I've heard of reintroduction efforts in the East Coast.
Yeah, I mean, in this state, people get paid to trap beavers.
Tara, thanks for being here. Would you speak to the status of our pika population and how it fits or doesn't fit with the narrative about pikas in peril?
Yeah. So pikas. So we had a volunteer that started monitoring pikas. I was recently corrected that the plural of pika is pikas and not, yeah, but whatever. Um, so we have amassed an enormous amount of data about our pikas, and we have not had the support or the funding to analyze it. And that's another another place where I have been trying to get funding, and I think that we're getting closer and closer to it. So far, we just count them, and so I can't speak to their status or their change in their status in the 12 years that we've been counting them, but they appear to be abundant, and they appear to be in a lot of great habitat, and they also appear to be in a lot of really, really bad habitat, which suggests that in like wildlife language, they would call that a surplus. And so the idea is that the young or inexperienced animals are being pushed into this marginal habitat, like underneath the warehouse, and that is often an indication that the populations are doing well. One of the things that we're working on is doing a lot of data cleanup so that we can pass it on to researchers that we're hoping to collaborate with at Washington State University so that they can analyze it for us and also help us redesign the project so that we can be better set up for volunteer involvement, because a lot of the sites that we have are really treacherous, and some of them, the ones that are easy to access, are along roads, where people can get hit by cars. And so we did a little bit of pilot work just to kind of like see like how could we integrate volunteers into our pica surveys last season, and what it kind of revealed is that we need to be a lot more thoughtful, and we need to have clear protocols and plans before sending people out, so that they're successful and we don't scare them.
I had two-part question, I guess two questions. About two different animals. First one, the invasive species of banana slugs.
Yeah.
I'd like any information about that. And then second animal was salamanders, and if there was any opportunity throughout the summer to conduct any research involving them.
Mm-hmm. So we detected there's a like pretty much all of the yellow or mustard colored or like baby poop colored slugs in the park are either banana slugs or leopard slugs, and they're both native. The ones that are not are smallish in like, they're usually not longer than like your finger when they're fully stretched out, and they're solid black. The tricky thing is that there are also some variations of banana slugs and leopard slugs that can be really dark in color. But we detected them for the first time, or at least recorded them during our BioBlitz in 2016. And so we didn't do a comprehensive survey of the whole park, but we did find them at Sunshine Point. We found them along the roadsides, and I don't have a good sense of how widespread they are, but when I see them, my partner just yelled at me for doing this yesterday, but I kind of just pretend like I'm Godzilla and like smash them. It makes me feel better. It's not going to do anything to their population, but that's what I do with them. And in terms of amphibians, Rebecca Lofgren and Scott Anderson are probably the best people to talk to about that. We do have a community-based science program, but I don't know how they're planning to pivot due to COVID.
Awesome. Thank you. And then I just thought of this too. Last year I saw a pig go into the forest.
I know, I want to know where that was.
Yeah. And I was just wondering if like there's been anything else that has been happening regarding pigs in Rainier.
No, but that's a really big deal.
Yeah.
I mean, because there are, I mean, in Oregon, in like, I think in Southwest Oregon and in Northern California, they do have problems with feral pigs. Um, and so like whether it was like an escaped pig from a farm or not, like they all have the capacity to become feral. And so, if it's a female, they have massive litters. And so, yeah, it's a big deal. And like, I would probably, like, if you can remember, like, and pinpoint where you saw it, you can put it in the Wildlife Observations database, but we can also report it to the Invasive Species Council, because they may have more resources to follow up on that.
Okay, thank you.
Yeah.
So, I have a question. I know that you mentioned the Ohana folks could possibly help out with your bat colony emergence monitoring. Is there anything us Sunrise folks could be on the lookout for in terms of like helping you guys out, volunteering where we can?
That's a good question. We don't have any known bat colonies in the Sunrise area.
Mhm.
And I think so. And so in terms of bats, I think the best opportunities to help with emergence counts are at Ohana, at T Woods, and at Longmire. Um, but our collaborator Jocelyn Akins is working on identifying the priority areas for us to survey for foxes this season.
Okay.
And we definitely have foxes up at Sunrise, and so there may be opportunities to help with some of those trail surveys.
Yeah, definitely.
Tara, could you talk to us a little bit about the impact of dogs on wildlife in the park?
Yeah. There are a lot of people with fake service animals that I reserve a very high level of judgment towards, with no apologies. I think those people limit access to people that have legitimate service dogs, and they also um, have lots of different types of impacts on wildlife, even if they're leashed. When they, carnivores communicate a lot of really explicit information by pooping and peeing everywhere. That's how they define their territories. And when domestic dogs, poo and pee, even if you pick it up, there's still residual scents that may affect animal behavior, particularly wide-ranging species like wolverines. It may cause them to avoid areas. We can't really say for sure because we haven't done experiments with that, but if domestic dogs are a sign of human presence, then having those scents left behind could certainly influence them. Like when we are setting up our cameras for wolverines, we don't eat in the area, we don't pee in the area. We make sure to limit any scents that we leave behind, so we minimize our influence on their behavior. And the same thing with foxes. So foxes and dogs are both canids, and and coyotes, and they can harbor all of the same parasites. And even dogs that are well vaccinated, they can carry worms when people are traveling across the country. We don't have a high prevalence of heartworm here, but in other parts of the country they do. And if people aren't treating their dogs and cats, for that matter, I mean, there are people that travel with cats, for parasites, or if they're not keeping up on their vaccinations, dogs can be asymptomatic for parvo and distemper. and cats can, they carry feline leukemia and FIV, which is similar to HIV, and that's a big problem that you see in feral cat colonies and places where they support trap, neuter, release programs. Is that they're serious disease vectors, and so that's something that I'm concerned about. And I don't have a lot of empathy for people that want to take their dogs in parks. It's like, go to the Forest Service lands. Like, can't we just have this one space, please?
I have a follow-up question to that. I may be wrong in thinking this. I think I had read it somewhere, but if you do have like your pet on any sort of like parasite control, like a flea tick medication, the fur that has those oils with the essentially like their medication on it, if that enters like the ecosystem and birds use it for their nests and things, I've heard that can be very detrimental to birds who are particularly sensitive to, like DDT and other sorts of like, parasite prevention, I guess, or insecticides or anything like that.
Yeah, that's a good question. I mean, I've done some work with like residual pesticides in aquatic systems, and the only place where we detected, I'm forgetting the name of the compound that's in Frontline, fipronil is what it is. The only place where we detected that was in a pond immediately after we saw a dog jump in and swim. And there were other places where we surveyed that I'm certain they, dogs swim in those areas, but we didn't detect it. Not to say that it's not there or its degradates aren't there, because, there are a lot of compounds, like glyphosate is a great example, where it degrades fairly quickly, but its degradate product, which is called AMPA, doesn't. And so it just depends on kind of the screening that you do for different compounds. But I can't speak to how it might affect birds, other than maybe reducing parasites in their nests, if there was and that could potentially be beneficial.
Right.
But I just again lost track of time, and I need to jump off. But if you are interested, I can join you again for another chat. I don't want to rush, and I'm happy to just be a resource for everyone.
Thank you, Sarah.
Awesome. Thank you, guys.
Gracias.
Okay. Bye.
Bye.
No, oh my gosh.